The last video left off looking at monocot stems and the arrangement of tissues. So here's just a reminder, right? We have the vascular bundles that are arranged in these sort of loose concentric rings, but we don't have any solid vascular tissue system. So we just have ground tissue surrounding the vascular bundles and the epidermis on the outside. Compare that to a Untitled. So this is a Untitled stem in primary growth. We have the epidermis on the outside, just like we did with the monocot and everything else. We'll always have epidermis on the outside, except when we get to secondary growth. But here we have the vascular bundles arranged in this distinct ring. So even though there's space between them, we're still going to differentiate between the tissues formed by the ground meristem. So now we have pith in the middle, and we have cortex just on the outside here of these vascular bundles. There's not a lot of cortex here, not like in the root. So more pith, less cortex. We have this ring of vascular bundles, and we can get a close-up on that ring to kind of see what's going on. So here we have the epidermis, this outermost layer. It might have some guard cells flanking stomata. We might also find trichomes, specialized cells that we'd see in the epidermis. Right here you can see this cluster of cells with these unevenly thickened cell walls. Those are Collenchyma cells. They're often located in the cortex of young growing plants, such as ones that would be in primary growth. Other things that you might see here would be the thick layer of wax that covers the outside of many plants. So that's the cuticle. You would find a cuticle on basically all plants, but it'll be quite thick in some and really thin in others. So with the epidermis, the cortex just underneath that, which is formed by the ground meristem, and then we have all of these vascular bundles arranged in a ring. So this vascular bundle here, we have a layer of phloem fibers. So remember that the phloem is going to be on the outside toward the epidermis, and the xylem is going to be on the inside toward the pith. So vascular bundle, but it doesn't have any sort of bundle sheath around it. Instead, we have these phloem fibers that form next to the parenchyma cells in the phloem that give that some structural support. So they have really thick walls. They're sclerenchyma cells, long fibers that I think are technically actually part of the cortex. So we have that cap here. And then underneath that, we would find our phloem cells, which we made up of sieve tube elements and companion cells. Then look at this ring here, a continuous ring of hamburger-shaped cells, right? That's because they're small cells that keep dividing in two directions. They're dividing to the inside and to the outside. To the inside here, they're making xylem. To the outside, they're making phloem. So that is our procambium, which we're going to refer to as these two different parts. So there's procambium that's within the vascular bundle. We call that Fascicular Cambium because that bundle is also called a fascicle. So our Fascicular Cambium, which you might recognize that word from when we talked about pines and their needles being in fascicles, little bundles. So Fascicular Cambium is the procambium that's within the vascular bundle. We also have Interfascicular Cambium. Inter means between, so between our fascicles, we have some cambium. So Interfascicular Cambium is located between the bundles. Fascicular Cambium is located within the bundles. It's all the same layer of procambium that's now making our primary xylem and phloem. However, it'll have different roles when we get to secondary growth, and that's why we have names for it. So these regions where the interfascicular cambium is located are called the Pith Rays. So there's some weird stuff going on with what's making what, but just know the names of these different tissues. We have Pith Rays that radiate out from the pith, right? So here's our pith, has these Pith Rays that come out between the vascular bundles that connect to the cortex, and outside the cortex is the epidermis, our outermost layer. Within that epidermis, we're going to have specialized cells like guard cells that flank stomata and trichomes that are hairs in the epidermis. Our vascular bundles will always be producing xylem to the inside and phloem to the outside. Within those, we'll have some residual fascicular cambium. Between our vascular bundles, we'll have interfascicular cambium. And then I think I totally forgot to talk about primary xylem, but here it is. Those large vessel elements that always help you identify where the xylem is. And it's always toward the inside. Okay, so let's look at some differences in what's going to happen when we go into secondary growth. So here, in our roots, the cortex didn't make anything. The cortex got sloughed off. Now our cortex is going to be tissue that helps us generate cork cambium. So that cork cambium before, in the roots, was made by the pericycle. Now it's going to be made by the cortex. We're also going to have some roles for the interfascicular cambium. Oh, that I guess is actually produced by the Pith Rays. Maybe it's not produced from the procambium at all. That would make sense. Forgot about that. So our pith is in the middle, and then our Pith Rays are going to make interfascicular cambium, which is continuous with the fascicular cambium. We're going to have some procambium left over between the primary xylem and primary phloem. That's called our fascicular cambium. Those two together make a continuous ring that in secondary growth becomes the vascular cambium. Whew. Okay, so we'll talk about this more in our secondary growth lecture. But the cork cambium is going to make cork to the outside of the plant and phelloderm to the inside. Those three things together, the cork cambium, and phelloderm, are called the periderm. The vascular cambium is going to make secondary xylem to the inside and secondary phloem to the outside. And we'll go over all of the names for all of those things for the secondary growth lecture. But just for you thinking about the organization of all these tissues and what they do, now you know. But we'll go over it in more detail soon. Okay, I think I will stop there and do this next part in a new video, since that was a dense amount of content. And now we're going to shift gears.